Today I'll be talking to you about introduction to mycology, and we'll be talking mostly about the general mycology and just an overview of fungal infections. Mycology actually comes from a Greek word mycos, which means fungus. So lately, have you been aware of importance of mycology or fungus during COVID? Has anyone got any comment on that? Does anyone want to comment on the importance of fungal diagnosis during COVID? Yes?
Black fungus became prominent again.
See, yeah, very good. But I would like to improve on that. That's enough. It was okay that he spoke. The big thing is that I'm sure most of you knew it but don't want to speak in the group. The thing is, black fungus is a very layman term, and you all are now learning microbiology, so you will have to learn the scientific names also. I'm sure now onwards you will talk about this fungus in more. So black fungus, we were calling it black fungus because the lesions were black, not because the fungus was black. Okay, the lesions which it produced, the clinical symptoms which it produced were black in color. Please, I request you again and again, please don't talk among yourselves. If you have any query, please stand up. I'll give you a chance to speak. Mycology is a part of microbiology because it's a living thing, eukaryote, which can be identified only under a microscope. So it is a part of microbiology, and mycology deals with fungus. Till now we had been talking about bacteria, but now I am talking about fungus, which includes yeast and molds. This thing deals with the etiology, pathogenesis, diagnosis, treatment and prevention of fungal infections in human beings. Fungus is a ubiquitous organism. So we are talking only about those which have infection, which cause infections in humans. Now, this has gained importance due to the opportunistic and some fatal infections, generally in immunocompromised, especially now in this era. Infections in immunocompromised patients have become important because of more number of people who are immunocompromised, because of increased life expectancy. Then chemotherapy, which is going on for patients who have been diagnosed for cancer, and immunosuppressants in people who have immunological diseases or organ transplants. Then antibiotics and steroids are also being administered more than what they were being done before. Eukaryotes, these fungus are eukaryotes. They are included in eukaryotes. Where are the bacteria placed in classification? They are prokaryotes. Fungus is eukaryote because it has a true nucleus. They are heterotrophs. They do not have chlorophyll. Plants have chlorophyll, but these fungi do not have chlorophyll. They produce filamentous structures called hyphae. They produce spores, which can be sexual and asexual forms of reproduction. They are saprophytic, which means they grow on dead tissue, or they can be parasitic. Parasitic means that they will be growing on living tissue, living organisms, or even some type of living tissue. All fungi require organic source of carbon associated with decaying matter. Generally, the living cells have enough immunity and they secrete enough secretions not to allow the fungus to grow, but generally in dead and decaying matter the fungus can grow. They vary in complexity and size, ranging from a single cell microscopic yeasts to multicellular fungi or a combination of both, which are classified as dimorphic fungi. The cell wall of fungi consists of chitin and beta-glucan. Beta-glucan is now used for diagnosis of fungus, and both are polysaccharides and site of action of some of the antifungal drugs. Cell membrane consists of ergosterol. Sterols are the components of cell membrane, and in fungus the sterols which are present are ergosterol rather than cholesterol like that of bacterial cell. Ergosterol is also a site of action of antifungal drugs, that is amphotericin B and azole group of antifungal drugs. Most fungi are obligate aerobes. They will not grow in the absence of oxygen. They are obligate aerobes, and some of them are facultative anaerobes. Classification of fungi can be on the basis of their morphological forms, the type of infection which they produce, the site of infection, and type of sporulation. Yeasts are one of the form of the fungi which is classified on the basis of morphology. These are unicellular fungi. They are round or oval bodies which reproduce by formation of buds known as blastospores. You will have to learn this nomenclature like blastospores, keep in mind, and we'll talk about it in the later part of the lecture. Now on culture, they produce smooth creamy colonies. For example, Cryptococcus neoformans, which is a capsulated yeast. Because of the capsule, it produces a creamy smooth colony. Now first was the yeast. Based on the morphological characteristics, the fungi have been divided. First, the unicellular ones were called yeast, then the other are yeast-like fungi. They grow partly as yeast and partly as elongated cells. This form of undetached budding yeast cells appear as broad septate hyphae and are called pseudo-hyphae. They are not actual hyphae, they are pseudo-hyphae. The example of this class yeast-like fungi is Candida albicans. The third form is molds or filamentous fungi. They form true mycelia and reproduce by formation of different type of spores. They have vegetative aerial hyphae, and the examples are Rhizopus and Mucor. So Mucor was the fungus which was mostly identified during COVID. This is where it is. It is the filamentous fungi which grows as molds. Then there can be dimorphic fungi. Dimorphic fungi can occur in both the forms, that is molds as well as yeast. Yeasts were single celled, molds were hyphae and multicellular. But dimorphic fungi can have both the forms. As you can see, some saprophytic in this these dimorphic fungi, they have their mold form at 25 degrees centigrade. Generally it grows in soil, and the yeasts are growing at 37 degrees centigrade in host tissue. And you can see that there is a difference. At 25 degree you can see there are hyphae growing, whereas the these parasitic phase, which is at 37 degree, only the budding yeast cells or the single cells are seen to grow. Dimorphic fungi, this is the most of the pathogenic fungi which cause systemic infections are dimorphic. The examples are Histoplasma capsulatum, Blastomyces dermatidis, Paracoccidioiditis, then Coccidioiditis, then Penicillium arniffi and Sporothrix. These are the examples of dimorphic fungi. Vegetative structures of fungi. These are formed by the vegetative mycelia. Vegetative mycelia are the modification of single vegetative cells or hyphae. They have no reproductive value. The reproductive value is of the spores. The hyphae do not cause the species to propagate. They are important in differentiation and identification of clinically significant fungi. So they have special type of hyphae. Some fungus, they have special type of hyphae which helps us to identify their types. As you can see, there can be a nodular organ, or they can be racket shaped. It can be a favic chandelier, or it can be a pectinate body. Like these are the different forms of the vegetative form of the fungus which helps us to identify the species of fungus. There can be spiral hyphae as seen in the central part. Now, reproduction in fungi can be sexual or asexual. Sexual reproduction occurs by formation of zygospores, ascospores or basidiospores. Asexual reproduction can be with the help of budding or fission. You know the difference between budding and fission? Yes? Can anyone tell the difference in budding and fission? This is such a literal word, budding and fission. हाँ बोलो, खड़े होके बोलो.
बर्डिंग के अंदर जो पेरेंट होता है,
हाँ,
उसका वैक्यूम रहता है, और न्यू जनरेशन की अलग ही रहती है. फिशन में पेरेंट बॉडी ही डिवाइड
होता है. Very good. So this is how you can describe that budding. The bud comes out of the hyphae, and in fission the hyphae is divided, or the cell is divided. In asexual, spores are formed on or in the specialized structure. These specialized structure and spores, they can vary in size, shape and color, and these characteristics of the spores and the sporangium or the budding, they are constant for a particular species. So with the help of these spores and sporing bodies, we can identify the fungus. Now, asexual fungal spores, they can be of three types. Please don't talk. If you have something so important, you can leave the class. constant murmuring in the class doesn't help. So asexual fungal spores are called vegetative spores. They are of three types. One is arthrospores. They are formed by segmentation and condensation of the hyphae. The hyphae divides and then the cytoplasm condenses to form the spores. So the example is trichosporon, as you can see in the top photo. Then the other type is chlamydospore. These are formed within the hyphae. The hyphae does not divide and condense the cytoplasm, but the spores develop within the hyphae. The example is Candida albicans. These are thick-walled resting spores. Blastospore is a bud coming off from the parent cell. This is also seen in Candida albicans. I am giving the example of Candida albicans on and off because it is an important opportunistic pathogen which everyone has to deal with. So these three are the vegetative spores of asexual fungal spores. Asexual fungal spores, they are aerial spores also. These were with the hyphae. Now there are aerial spores also which will come out of the mold. That is sporangiospore. In this, hundreds of the spores are formed within a sac. The sac is called a sporangium. This sporangium is found at the end of an aerial hyphae. It is not developing. The spores are not developing inside the hyphae. There is a special branch of the hyphae which is called sporangiospore, on which the sporangium develops, and the examples of this is Rhizopus and Mucor. These two fungus have been very important in immunocompromised patients. Asexual fungal spores can be aerial spores. Conidiospore is one of them. These are multiple. They are formed in chains or single spores formed at the end of an aerial hyphae which is not enclosed within a sac. Just now I told you sporangiospores, they were enclosed in a sporangium. It was a sac in which the spores were formed. Here in conidiospores, the spores are in chains, but they are not covered in a sporangium. The examples are Aspergillus and Penicillium. These two fungus are important contaminants of the lab, and they can cause opportunistic infection. There are two types of aerial spores. One is Microconidia and the other are Macroconidia. In Microconidia there are small single cells. Macroconidia, there are large single or many spores called dermatophytes. Systemic classification of fungus. This is based on sexual and asexual reproduction. There are 4 classes. Based on sexual and asexual reproduction, there are 4 classes. One is Zygomycetes, Ascomycetes and Basidiomycetes. All these 3 classes, they reproduce sexually, whereas one class, that is Deuteromycetes, it reproduces asexually. Most fungi of medical importance lie in Deuteromycetes. Zygomycetes, these are the lower class of fungi. Here the hyphae is broad and non-septate. You understand what is a septa? Septa तो समझते हैं? हाँ या नहीं? Yes? Okay. So these are broad non-septate hyphae. They have asexual spores, sporangiospores, which are present within a swollen sac like structure called sporangium. As you can see it here in the figure. Sexual spores, zygospores, which is a resting thick walled cell in between the hyphae. You can see it in the second picture. The examples are Rhizopus, Mucor and Absidia. In Rhizopus, you can see the rhizoids. These are root-like structures which you can see down. That is its difference from Mucor. Mucor and Rhizopus, they differ with the presence of rhizoids. These are roots. Then second is Ascomycetes. These includes both yeast and filamentous fungi. These are narrow, those were broad. Zygomycetes was broad, non-septate. Ascomycetes has narrow septate hyphae. The asexual spores are called conidia. These are borne on conidiophores. The hyphae which bears the conidia is called conidiophore. In that it was known as sporangiophore because it was having a sporangium. Now sexual spores called ascospores are present within a sac. Ascus, you know, is a sac. So these are ascospores. They are present in a sac-like structure called ascus, and several asci may be seen within a fruiting body as seen in Penicillium and Aspergillus. Each ascus has 4 to 8 ascospores, as you can see in the figure. Third is Basidiomycetes. Sexual fusion results in the formation of a club-shaped organ called basidium, which bears the spores called basidiospores. You can see it in the figure. First is the basidium formation, then the nuclear fusion occurs, then meiosis occurs, and basidiospores are formed and then liberated. This is the sexual type of reproduction. These are the sexual spores, basidiospores, ascospore and zygospore, as you have already seen in different slides. The fourth is Deuteromycetes or Fungi imperfecti. These are group of fungi whose sexual phases are not identified. It is not that these fungi are imperfect, but lot of research has not been done on them, or even if the research has been done, a lot is unknown about these fungi. So they are known as fungi imperfecti. They grow as molds as well as yeast, both the forms. They are dimorphic. The asexual stage is conidia. Example is again Candida plus Cryptococcus also. Cryptococcus is an important fungi, causes encephalitis, meningitis, etc. Now the miscellaneous terms which should be known when you learn about fungus is conidiophore, which is a specialized branch of hyphae on which conidia are developed. Sporangiophore is a special aerial hyphae or stalk bearing a sporangium. Where it comes, the 4 comes in the end of the word, it means it is a stalk. It is a branch or a hyphae which is bearing conidia or sporangia. As you can see, these are the pictures of various fungi. Conidiophore is seen in that one, and the upper part is the sterigmata and vesicle on which the sterigma are seen and chains of conidia are seen. The conidia are youngest towards the sterigma and oldest at the end. Whereas in this you can see the rhizoid, sporangium, sporangiophore and spores in the first picture. Now miscellaneous terms, again sporangium, which is a sac, Demeticious or Phaeoid, these are pigmented, dark in color, usually grey to black. Sterigmata is a specialized structure that arises from a basidium and supports basidiospores. Other terms are hyphae, which are elongation of apical cells which produce a tubular thread-like structure called hyphae. Pseudohyphae is a chain of elongated budding cells that have failed to detach. There is a difference between pseudohyphae and hyphae. Hyphae are generally seen in molds, whereas pseudohyphae are seen in yeast forms. The yeast-like fungi, they produce pseudohyphae. Then rhizoids are root-like structures. Myceliums are tangled mass of hyphae. Fungi which produce mycelia are called molds or filamentous fungi, and hyphae may be separated into septate and non-septate as I was describing in the previous slides. Now mycotic diseases, there are 4 types of diseases produced by fungus. One is allergy, that is the hypersensitivity due to the presence of fungal elements on the skin or inhalation. Then there is mycotoxicosis, which is produced because of the toxins produced by the fungi. A person can have a toxic response to the mycotoxins. Then mycetismus, this is a preformed toxin which is generally seen in mushroom poisoning. When there are some mushrooms which are eatable and there are some which are not eatable, those which are not good for consumption are because they produce some toxins, and these toxins are preformed in the mushrooms. They produce a disease which is mycetismus. Then naturally these are microorganisms, fungus are microorganisms, they can cause infection. Now, categories of systemic diseases produced by fungus, those caused by truly pathogenic fungi with the ability to cause disease in the normal human host. When the inoculum is of sufficient size, these are truly pathogenic fungi. The examples are Histoplasma capsulatum, Blastomycetes, Coccidioides immitis, then Paracoxodioides brasiliensis, and those caused by opportunistic fungi. These infections occur when the immunity of the person is low. These are fungus with low virulence. They require the patient's defenses to be lowered before the infection is established. As I told you, Aspergillus and Penicillium, these are generally lab contaminants. But if the person has low immunity, if a person is working in the lab and he has low immunity, he can acquire infection because of Aspergillus. Generally, I am sure you all have seen Aspergillus growing on bread, moist, organic items which are left in the kitchen or elsewhere. In your gardens also, you have definitely seen green and black fungus. So these can cause infection if you have some immunocompromised person in your family. Please take care. Fungal infections are difficult to treat. Okay. Now, Cryptococcus neoformans is a very deadly disease caused by fungus. Now there can be fungal infections or mycosis. This can be superficial mycosis. Superficial, you understand, it is on the surface of the body. These can be of two types, surface and cutaneous mycosis. It can be on the surface or it can be on the lower part of the skin. It can be in skin, hair and nails. These are the surface structures. It can be skin, it can be hair or it can be nail. So this superficial mycosis can be seen in all the three surface structures. Then there can be deep mycosis, which can be of two types. It can be subcutaneous and systemic as well. Deep mycosis will include subcutaneous, whereas deep mycosis, it ranges from a symptomatic infection to fatal disease. Then there can be opportunistic infections. Mostly in opportunistic infections, it's a systemic involvement. Now, topographic grouping of fungi. Superficial is surface infection which is confined to the outermost layers of the skin, hair and nails. No host cellular or inflammatory response is seen because the skin and hair, they have a dead tissue on the outside. This disease is recognized purely on the cosmetic basis. Have you seen any Pedra disease? Generally लोग बाग उसको ringworm type कहते हैं, but it's a fungal infection. Pedra diseases होती है, skin का discoloration होता है, chest पे generally boys have it. They have it on the chest and neck. Anyway, this is a cosmetic importance. Then it can be cutaneous. Cutaneous means that the keratin of the skin, nails and hair is involved. These organisms prefer non-living cornified layers. They go in the skin, but they are only limited to the non-living cornified layer of the skin. The disease is called dermatophytosis. Please mark, this disease is called dermatophytosis when caused by dermatophytes, or it can be called dermatomycosis when it is caused by candida, or it is a cutaneous manifestation of a systemic disease. So there is a difference in dermatophytosis and dermatomycosis. Now, host response in superficial this thing is patchy scaling or eczema type eruption. This is mycosis, superficial surface skin, hair and nails, mild but chronic disease. They live exclusively on dead surface of the skin and its appendages. There is no contact with the living tissue, hence no inflammatory response. The diseases are Tinea versicolor, Tinea nigra and Tinea pedra. All this is on the skin, hair and nails. In this, infection is in the conified layer of the skin and its appendages. The presence of fungi and its metabolic products, they cause inflammatory and allergic responses. This is cutaneous. Then dermatophytes, these can be of three types: trichophyton, microsporum and epidermophyton. These three are dermatophytes which are important. Some cause infection in the skin and nails, some cause infection in the skin and hair, some cause infection in the hair and nails only. So Subcutaneous involve the deeper layers of the skin and also the muscle tissue. In this case, man is an accidental host following inoculation of fungal spores via some form of trauma. This type of infection is often identified by the presence of a characteristic tissue reaction or granule is formed. In the cutaneous, there was no reaction, but here there is tissue reaction and granule formation. And then the last is systemic. This attacks the deep tissues and organ systems, even organs like lung, liver, kidneys, all can be involved. And these create symptoms that resemble other diseases. It's very difficult to identify a fungal systemic infection. Then these are caused by soil saprophytes or plant pathogens. In this case, infection is accidental, the risk factor is trauma, localized, it can be localized or may spread by conguity. It can spread from the site of infection to other parts of the body in conguition. It will not go through blood. Ranges from a symptomatic infection to fatal disease. The examples are mycotic mycetoma, chromoblastomycosis, sporotrichosis, rhinosporidiosis, and subcutaneous phycomycosis. You can see the subcutaneous. Generally, there are eruptions on the skin which have the fungal tissue, which you have to take on a slide and identify. There may be granules like sulfur granules which can identify actinomycetes, etc. This can also be caused by actinomycetes. Now, deep mycosis caused mostly by soil saprophytes. Most of the fungus are soil saprophytes. Dimorphic fungi, and these can vary in the degree of severity. The examples I have already quoted, and these are the features of the systemic mycosis. They can be inhaled, then they will cause symptoms in the lung. They can have flu-like symptoms. They can go to lung where it can cause consolidation, and they can mimic tuberculosis. They can go in liver, bones, etc., and there they will mimic all the organ diseases, and it is difficult to identify the fungal infections. Now, candidiasis is caused by Candida species. Candida forms a bridge between superficial and deep mycosis. This is caused by Candida species, which forms a bridge between superficial and deep mycosis, as it can cause cutaneous as well as systemic infections. This Candida can also cause opportunistic infections. Opportunistic infections occur in patients with debilitating diseases, altered physiological state, mainly caused by fungi, which are common lab contaminants on culture media. They produce serious and fatal infections. The examples are Aspergillus, Penicillium, Euchre, and Rhizopus. The diagnosis of fungal infection and Candida is mostly based on microscopy, where you take a wet mount of the sample, you put it in KOH. KOH is, what is KOH? Potassium hydroxide. It helps to dissolve the tissue, and it will leave the fungal elements as is, so you can see them under a microscope. Then there can be skin test, which is known as dermal hypersensitivity test. You can inject the antigen and you can see the hypersensitivity reaction. Then there can be serological test, which means you take the blood, take out the serum and test for antibodies of the fungus. Then there can be fluorescent antibody test. In this you use a fluorescent microscope, use a fluorescent dye and tag it with the antigen so that this goes and binds with the antibody and gives a fluorescent appearance under the fluorescent microscope. You can take biopsies from the tissue and see them under the microscope to see the granulomatous or necrotic tissue and the specialized structures of the fungus like spores, sporangium and conidia, blastospores, etc. The fungus can be cultured on special media, which is generally the Sevrod's dextrose agar. This media fungus needs a bit of acidic pH to grow, and antibiotics are added in the media so that the media is not contaminated by fungus. For fungus culture, the media is to be kept for 2-3 weeks. So there is chances of bacterial contamination, hence antibiotics are added. And lastly but not the least, DNA probes, which means molecular diagnosis of fungus, is also important. Epidemiology is important for fungus because it is generally a saprophytic environment where it is present. It is generally present in soil, so they will have special type of fungus in different areas. It may not be present, all the fungus may not be present in all the areas. And Candida albicans is a part of normal human flora. As I said, Candida albicans is an opportunistic infection because it is a normal inhabitant. As soon as the immune status of the person is below a level, this normal inhabitant will get an opportunity to cause a serious infection. Most mycotic agents are soil saprophytes. Mycotic diseases are not contiguous except in few cases as in superficial mycosis, which means they are not acquired by contact. Fungal infections do not spread by contact. The establishment of infection depends on the inoculum size, how much the fungus is inoculated for infection. The resistance of the host is important rather than the virulence of the fungus. If the fungus is very virulent but the immune status of the person is good, he will not acquire the infection. But if the virulence of the fungus is low and the immunity of the person is less or decreased, then he can then also acquire infection with fungus which is very low virulence. The useful properties of fungus, as you can see, they are a good source of food. For example, mushrooms and broccoli, they provide all the amino acids which are needed for human proteins. Okay, how many amino acids are needed for human growth? Amino acids. Anyway, this is not out of subject, but you must know that fungus, mushroom is very important source of amino acids for human proteins. Then fermentation, production of alcohol and bread because of Saccharomyces. This is a fungus which causes fermentation for production of alcohol and also in bread. There is fermentation in bread also. Then antibiotic production can be from the fungi. For example, penicillium. Penicillin, you all know. Penicillin is used for streptococcal infections. So, it is acquired from Penicillium neonatum. Then other useful properties are ergot from Claviceps purpurae is used to induce uterine contractions. During labor, if the uterine contractions are not happening and not proper, ergot was given, though it was given a long time back. Now it is not given as ergot, but other medicines, they can also have ergot. Then vaccines for hepatitis B can be formed by Saccharomyces cerevisiae. I think this is all for today. So